Hello, my name's Tim, and I'm a business development specialist with Synbio Technologies, and today I'm here to talk with you about gene synthesis. Quick overview for gene synthesis is first we'll start with your sequence of interest, then we'll design oligos to cover that entire sequence. We'll use PCR to assemble those oligos into larger gene fragments, and take those gene fragments and clone them into the vector of your choice. We'll perform some quality control steps to make sure the sequence is 100% correct, and then we'll deliver that final product to you. So now let's take a deeper dive into each one of these steps. For step 1, sequence determination, you can deliver your data in 3 different formats: the DNA sequence itself, amino acid sequence, or the gene ID. We can take these sequences and input them into our proprietary codon optimization software. This will give us the optimal DNA sequence for your species of interest to maximize expression. Now we're ready for step 2, oligosynthesis. I'll have a video that covers this in greater detail in the link below. However, to give us a general overview, it's a 4-step process that can repeat that adds DNA nucleotides in a sequential manner. We can use this to design specific oligos that we can use in our later synthesis steps. Now that we have our oligos, we're ready for PCR assembly. Here in purple, we have the entire gene of interest, and you can see that the oligos between the complementary strands cover that entire gene of interest. They also have overlapping regions that allow them to anneal to one another, essentially creating primers for DNA polymerase to bind to. The DNA polymerase can bind to these pseudo-primers and fill in the gaps between the oligos. Then we can add primers on either side of the gene of interest that will amplify out our gene fragment. Now that we have our final product, we're ready to move on to our final step, cloning. Let's start with a basic overview for the cloning procedure. We have our 2 components, the vector and our gene fragment. We'll use a method to expose the ends on both the vector and the gene fragment. These ends will anneal together, and then we'll repair any damage to the DNA, such as nicks or missing bases. The 3 most common methods we use at Synbio are restriction enzyme and ligation, homologous recombination-based assembly, and golden gate assembly. For restriction enzyme and ligation, we add a restriction enzyme that cuts at a specific sequence. Between the gene and the vector, they both have the same or similar restriction enzyme sites. And so you can see here in blue, the restriction enzyme binds, and then in the red dotted line is where it's going to cut. When the restriction enzyme cuts, it creates 2 sticky ends. If there are similar restriction enzymes, they'll create sticky ends that are complementary to one another. So these ends can anneal, and then we can add a DNA ligase in order to repair any nicks that are created. For homologous recombination-based assembly, the ends of the vector and the gene fragment are homologous. So we can add a exonuclease that will degrade the DNA in a 5' to 3' direction, thus removing those bases. This will create sticky ends again that will be able to anneal to one another. However, that exonuclease is not specific, so it'll cut past the region of homology. This means we have to add in a DNA polymerase in order to fill in those missing bases, and then we can add a ligase in order to repair the nicks that are created. For golden gate assembly, it's going to use a restriction enzyme as well. However, it's a unique restriction enzyme. As you can see here, the restriction enzyme binding site, or recognition site, is outside the region where it'll actually perform the cut. So you can use one restriction enzyme to create multiple unique sticky ends. This is useful if you're cloning together multiple gene fragments in the same reaction. So here we have our restriction enzyme. It cuts, creating those sticky ends. They anneal to one another, and then we can use again a ligase to repair the nicks that are generated. With cloning completed, we're ready for sequence validation. The 2 methods we use are Sanger sequencing and gel verification. Sanger sequencing will be performed to make sure that the gene fragment that was cloned in the vector of choice is 100% sequence verified. A restriction digest will be performed in order to make sure that the vector plus the gene fragment are of the correct size. So now that you know a little bit more about gene synthesis, what are some of the advantages of working with Synbio Technologies as your one-stop DNA service provider? Well, some of the key advantages are we have an experienced project management team that is capable of handling complex and difficult to synthesize sequences. We guarantee 100% sequence accuracy, and your intellectual property is always protected. So now let's look at some case studies where we had complex and/or long sequences to synthesize. Here's one that's only 580 base pairs, but it has 15 LOXP sites, creating a lot of secondary structure. We were able to successfully deliver this within 4 weeks. Again, we have one that's 1600 base pairs, but it has a high concentration of GC at 72% and 176 CpG residues. We were able to successfully synthesize this and deliver it within 5 weeks. And we're also capable of handling large fragments. So here's one that's almost 14,000 base pairs, and we were able to deliver this within 4 weeks. When you're ready to start your gene synthesis projects, there are several ways you can reach out to Synbio. The easiest is to create an account on our website, and then you'll have access to this quick order menu. Here you can upload your sequence and you'll get an instant price quote for your project. Another option is you can send us an email at quote@synbio-tech, and we'll get back to you within 24 hours with a price for your specific project. I hope you got something out of this presentation. My name's Tim, and I'm happy to answer any of your questions, so please feel free to email me at timothy.acri@synbio-tech.com, and I hope to hear from you soon. Thank you for your time, and I hope you have a wonderful rest of your day.